Topic of this video is what is microbiology and what are the applications in the field of microbiology, what are the basic procedures that we perform in microbiology. So let me give you an overview for field of microbiology. Microbiology basically consists of microorganisms which include viruses, fungi, and even parasites. But we mostly do conduct study related to bacteria. Microbiology is very diverse field. It includes literally everything, and bacteria are found everywhere. They are in soil, water, plants, everywhere. Soil is known as reservoir of bacteria. Your body has bacteria. Air we breathe can have bacteria due to aerosols. There's no place where there are no bacteria. You can find them in atmosphere as well. So there you can find them in deep ocean vents. You can find them in ice. Similarly, fungi are also quite diverse. They have multiple applications. Now let's come to the main point, that how do we conduct research, or what are the basics of microbiology which we really need to know in order to study the subject of microbiology or applied microbiology. Well, basically, first of all, you really need to understand the basic procedures we do in laboratory. For that, first of all, you need to know that personal protection is the most important part. I mean, you really need to wear gloves. You need to know about gloves. There are many types of gloves. Latex gloves, they are very helpful in preventing bacteria to harm you. You can use nitrile gloves. You can use vinyl gloves if you want chemical resistant gloves. If you are working for certain chemicals, using certain chemicals, you need to use chemical resistant gloves. Secondly, you need to use a lab coat so that you're safe from chemical spills or bacteria and all that. Third, you really need to uh, keep the environment clean where you are working, because the worst nightmare a microbiologist or any other field who is working with bacteria, the worst nightmare is contamination. So in order to avoid contamination, you really need to keep the place clean so that you don't have to face the problem of contamination. Contamination is a very big issue. Most of the time you are working on a single species of bacteria, for example E. coli or salmonella. But if you want to obtain, for example, you want to obtain salmonella, you really need to eliminate the contamination. I mean, when you take sample from environment, you need to kill all those bacteria you don't want on your media, so that you can specifically select that bacteria you want to have cultured, so that you can use it afterwards. So for that purpose we use autoclave. Mostly we use autoclave. We can use hot air oven, but most of the time we use autoclave. The reason is that autoclave has a high temperature, 121 degrees centigrade and all that. But the most important thing is that it creates a pressure, and pressure is created with the help of water vapors, and that pressure helps in killing most of the spores and bacteria. And when that is done, most of the contamination is over. I mean, the petri plates, there are circular petri plates that are used to culture bacteria. Test tubes, flask, and even media are now free of contamination. But in some cases, there are some substances which are, sterilized with the help of microfilters. Now, what are microfilters? Microfilters are basically small pore sized filters which help in removal of bacteria. There are some substances which can be damaged by heat. So these, for example, that those substances include urea. So urea can be damaged by heat and form ammonia. So in order to avoid that, what we do is we use microfilter instead of autoclave. So that is the most important thing you need to remember. Hot air oven can also be used. You can obtain a higher temperature, but we don't really use it. We don't really use hot air oven, but we can also use it. And there are several other methods by which we can sterilize workplace, like chemical compounds. They have different mechanisms. I mean, we can use phenols, formaldehydes, ethanol, spirit. We can use them. And we can also use UV to sterilize workplace. So coming to the next point, when we have prepared our media, now what is media? Media is a chemical complex chemical composition, basically, in which there are several factors, several things available which promote growth of bacteria or fungi. Even in the case of parasite, media is different. For example, for Naegleria growth we use E. coli, but for we are talking about bacteria only in this video. So media can be complex, selective, differential. The main thing is that for selective growth, for example, we want the growth of gram negative bacteria. So if you want gram negative bacteria, you need to eliminate the growth of gram positive, and for that we can use any kind of antibiotic, so that the growth of gram positive bacteria is stopped and gram negative bacteria can grow. We can also make media, for example, potato dextrose agar, which have a ability to promote growth of fungi only instead of bacteria. And we can also include dyes and different indicators which produce colored compounds. For example, EMB agar, eosin methylene blue, it will produce green metallic sheen when E. coli grows. If it is SS agar, Salmonella shigella agar, then black and white clunies can produce indicated bacteria, Salmonella shigella. Or even if it is blood agar, which is used for staphylococcus and all that. These media are basically medical related, so you don't really need to go in all of that. You can only know about nutrient agar and yeast extract. Those are the most commonly employed medias in the lab. And if you are working on certain conditions, then media is usually formulated on the basis of that purpose. I mean, it can be anything. If you are working on a fertilizer, biofertilizer, biopesticide, then those medias are customized according to that method. I mean, if you want to degrade plastic, then media must contain all sorts of minerals without carbon source so that bacteria can grow on the carbon source which is available in media, which is plastic. So there are different applications. I will come to that in another video. So here we must focus on what is the common procedure we perform in lab. Now let's move further. You must also know that laminar flow is also very important. Laminar flow, we usually work inside a laminar flow. Without working inside a laminar flow, you will get contamination again inside media when you incubate, or it can be also harmful for you as well. You must also stay safe from bacteria. So laminar flow basically consists of air cutting, and it can also consist of UV, and it is a safe environment in which you can work. So coming back to the topic, now let's say you have taken the samples. The sample taken must be sterile. I mean, you can use a cotton bud to take a sample, for example. That swab must be sterile. Now how will you obtain bacteria? Now first of all, you need to learn about agar. Agar is a gelatinous compound. I mean, it is a kind of a jelly which has a unique property. When you heat agar, it will become liquid, but when After heating, you let it cool down, it will become a solid, and you can use agar. And by adding something inside agar, it can be any kind of media. When you add media and agar, it will form a solid support in circle petri plates. And when you incubate, after incubation time, you will see certain white clunies, or if clunies can be of any type, I mean any color. So you will see different clunies forming up. Now how will you do that? You, after you have solidified media on petri plate, you can do three different methods. You can use streaking, you can use spreading, you can use pouring method. Pouring method is you're not usually employed, because in that pouring method, what we do is we put inoculum first, then we add media, and then let it cool down. So it is usually used for micro aerophile. But spreading method is different. In spreading method, we use a spreader to spread the bacteria, and you can see a good uniform colony afterward. In case of streaking, it is a very different technique, and it is mostly employed to obtain isolate. For streaking, we use inoculating loop, which is made from platinum, and it has a unique property. When it is heated, it turns hot, and when it cools down, it becomes the what it was before. I mean, brown colored and all that. It can be blackish, but it will help you identify that the temperature of inoculating loop is now cool. So now you can use it to inoculate, so that bacteria is not dead if you touch inoculating loop while it is hot. First, you add inoculating lube inside the sample, then you make lines from one side of the plate to the another, and after that you place the inoculating lube inside the burning flame and heat it. After heating, let it cool down, and now what you will do is, after that, make more lines from the point where last lines ended, and repeat it again until you will make lines in a almost square. The point is that these isolates will separate, and it will form separate colonies at the end. Microbiology, now coming to the applications. Microbiology has several applications. Microbiology includes, I mean, food microbiology. I mean, you can use probiotics, and you can find different health benefits due to those probiotics, and it is a very vast research. You can use probiotics to, for example, eliminate toxicity of heavy metals, or treat different types of pathogens and all that. You can also use different types of bacteriocins. You can use bacteriocins as well. And food microbiology is quite diverse subject. Another application is environmental microbiology, in which you can study impact of microbes in environment, from, for example, different types of cycles and all that. In bioremediation is a very important topic. Bioremediation includes detoxification of for example, heavy metals environment. You know that there are several heavy metals found in river, sea, and drinking water. You can use bacteria to detoxify those heavy metals, pesticides, and you can also, what we can do is that we can also do research on biocorrosion. Microbes can cause corrosion. We can find different ways to avoid corrosion, and we can use bacteria for plastic production. We can use bacteria to degrade plastics. We can use bacteria to make different types of dyes, pigment dyes, which are, for example, you can make paint, a paint which can be antifungal in property, or we can also work on different types of antibiotics, because we know antibiotic resistance is a very big issue. So we can work on this. Microflora is also a very important research topic. and microflora is present inside everyone, I mean humans, animals. So microflora is usually connected with diseases, so you can conduct research on that as well. So these are just some of the few applications of microbiology. We will discuss each one of them in detail in next videos. So please like, share and subscribe.